Okay, hello everybody. Thank you very much for joining this webinar. My name is Viviane Sternkopf and I'm your moderator for today. Together with my colleague, Kimberley Gutierrez, I would like to invite you into the very exciting topic of how crystal digital PCR can help you to investigate critical genetic marker in oncology and introduce you to the new 6-color NICA system from Stilla Technologies. Before we start with the presentation, I would like to introduce you to the widgets that will allow you to address questions to us already during the presentation. We will collect these questions arriving during the talk and try to answer as much as possible into the later question and answer section. If your questions might not be answered for time issues, we will get back to you after the presentation via the contact details provided with your registration. As you can see on the slide, you will find an option to enter your questions on the right side of the screen with enter your text, and then please hit the send button so we can receive your questions. The presenter of today's talk with the title Liquid Biopsy Target Quantification Using High Multiplexing Cancer Panel and Crystal Digital PCR is my dear colleague Kimberley Gutierrez, who is a Senior Field Application Scientist at Stilla Technologies, Inc., based on the US East Coast. Kimberley received her PhD in microbiology from the University of Washington in Seattle, where she studied viral host interactions of Kaposi sarcoma herpes virus infections. She completed a postdoc in immunology, investigating programmed cell death and activation of innate immune response. She previously held positions as microRNA application specialist and an NGS field application scientist before joining Stilla Technologies, Inc. in July 2019. As a field application scientist for Stilla, Kim serves as a scientific liaison supporting the technical needs and the customer to implement digital PCR as multiplexing approach to nucleic acid detection. With this, I would like to hand over to Kim and say, Kim, the stage is all yours.
Thank you very much, Viviane. So welcome everyone. Thank you for tuning in for our second installment of our June seminar series focused on our 6-color multiplexing technology. So I'll start with an overview of the talk today. I'll begin by presenting crystal digital PCR and the NICA system workflow and describe the current 3-color system and then introduce our new 6-color system. The second part of the talk will focus on 3 6-color crystal digital PCR cancer panels, a lung cancer panel for EGFR mutation detection and patient monitoring, and a breast and lung cancer panel, and rectal cancer panel, both currently being used in clinical studies. Stilla's NICA system is a unique and flexible multiplex platform to perform crystal digital PCR. The system consists of one consumable, the Sapphire chip, which holds 4 samples. The chip comes pre-filled with oil, so you simply load 25 microliters of your PCR master mix and cap the inlet port. The NICA geode performs both the partitioning of the sample and PCR amplification of targets within the Sapphire chip. 3 Sapphire chips can be loaded at one time in the geode for a total of 12 samples. Once the partitioning and amplification is complete, the chips are imaged and read in 3 colors, blue, green, and red, by the NICA Prism 3. And finally, data analysis is completed using our free and fully integrated software, Crystal Miner. Now that I've introduced the NICA system, I'd like to take a moment to describe the principles of digital PCR. In digital PCR, the workflow starts with the same reaction mix as for qPCR, containing the reagents necessary for the amplification of target nucleic acid. The key step in digital PCR is the partitioning of the reaction mix into a large number of small compartments of the same size. In the NICA system, partitioning is generated by a water and oil emulsion of the mix. As the emulsion is generated, the target sequences are randomly distributed in the droplets, such that some droplets contain one or at least one sequence of interest, and some don't contain any sequence at all. After partitioning, each droplet acts as an independent reactor where PCR can occur if the target is initially present. A successful PCR will lead to the generation of a fluorescent signal at the end of the reaction, and the fluorescence intensity of each droplet is then plotted via a dot plot with 2 clusters separated by a threshold automatically set by the analysis software. Above the threshold are the droplets considered as positive, and under the threshold are the droplets considered as negative. The analysis software then uses the proportion of positive droplets and Poisson statistics to estimate the concentration of the target of interest together with the uncertainty on the measurement. The latter lies in the fact that a positive droplet may contain more than one target sequence initially and is dependent on both the concentration of the target in the PCR mix and the number of droplets generated. The patented partitioning technology by contactless fluid injection in the NICA system leads to a self-assembled array of droplets called a droplet crystal. In a droplet crystal, the size of the droplets is fixed and controlled via the microfluidic design. Ensuring a monodispersed population of the droplet size is important in order to guarantee a random distribution of the target sequences within the droplet crystals. The other specificity of the NICA system is that it has 3 fluorescent channels, allowing for the quantification of 3 different targets in a unique sample. In this schematic, for example, 2 variant sequences of genomic DNA can be quantified together with the wild-type sequence. In such an experiment, the lowest abundant target, in here red, is artificially enriched by partitioning, making it less prone to competition, as would be the case in a bulk solution in qPCR. This is why digital PCR is more sensitive than qPCR to detect rare events. Making digital PCR a lab commodity is one of our missions at Stilla, and the NICA system provides an easy-to-use and integrated solution. There is a single pipetting step required to load the sapphire chip, making it robust against contamination and very easy to use. You then simply place the sapphire chips in the geode, which combines partitioning and thermal cycling. Once the PCR program is complete, you transfer the chips to the prism and scan the 3 chips in 3 colors in about 10 minutes or 50 seconds per well. And finally, you can analyze your results with our free and integrated software. In summary, the NICA system allows you a fast time to result in about 2.5 hours with minimal hands-on time. I'd now like to introduce Stilla's newest innovation, our 6-color crystal digital PCR. As you can see, the setup is similar to the 3-color system, with 1 pipetting step to load the sapphire chip, the NICA geode to perform the partitioning and thermal cycling. However, we have now doubled the multiplexing capability from 3 to 6 with our new 6-color reader. We also still have our fully integrated software that is contained within the workflow. 6-color crystal digital PCR uses a range of fluorophores, and listed here are some examples of compatible fluorophores. However, this is not an exhaustive list. The 6-color reader is compatible with the sapphire chip, which can run 12 samples at a time or 36 samples in a day. It is also compatible with our soon-to-be-released opal chip, which allows you to run 48 samples at a time or 144 samples in a day. You can still run 3 chips at a time with a time to result in under 3 hours for all 6 channels. The NICA system 3 and 6-color are compatible with a range of applications across life sciences and translational research. The remainder of this talk will highlight 3 different 6-color oncology panels developed by Stilla for patient monitoring. The first 6-color crystal digital PCR panel for oncology, developed by Stilla, was designed to quantitate EGFR mutations to monitor lung cancer. The main motivations for this project was to develop a single 6-color assay to simultaneously detect 90% of known EGFR mutations in non-small cell lung cancer, while reducing the time and cost for monitoring without compromising sensitivity. Some of the main challenges involved the need to monitor a large number of EGFR mutations from a single liquid biopsy blood sample to provide a quick answer and an adaptive treatment for the patients. We incorporated multiple strategies, including a drop-off assay for detection of insertions and deletions in exon 19, and the multiplexing allowed for the ability to differentiate EGFR TKI resistance mutations from sensitizing mutations. In the bottom right of the slide, you will see the panel that was developed to detect 90% of these known EGFR mutations in a single assay. You will notice that there are multiple mutations are grouped and detected by a single fluorophore. This was done based on the relevant clinical treatment, and an exogenous internal control was also used to monitor the efficiency of the reaction. This panel quantifies 19 of the most prevalent TKI sensitizing and resistance EGFR mutations and allows us to maximize the use of patient samples while minimizing the time to results. This work was published in Oncotarget in collaboration with Gustave Roussy in Paris. Both tumor and cell-free DNA samples were used to test the clinical utility of the 6-color EGFR panel. The circulating DNA samples were used to monitor EGFR mutation status over time from patients with non-small cell lung cancer. The change in EGFR sensitizing and resistance mutations correlated nicely with both disease state and the chemotherapeutic regimen. This initial study demonstrates the suitability of 6-color digital PCR to monitor patient response to treatment using a non-invasive liquid biopsy sample. I'll now move on to some work we have been doing in collaboration with the Horizon 2020 project on breast and rectal cancer. The major motivation for this study was to combine liquid biopsy monitoring and MRI scans to predict and monitor cancer therapy responses. This work is part of a consortium from the Horizon 2020 funded LIMA project led by Philips. The main challenges for this project is the number of samples we need to process. The study enlists 100 patients per trial, with 10 samples per patient for the breast cancer trial for a total of 1,000 samples, and 4 samples per patient for the rectal cancer trial for a total of 400 samples. The goal was to develop a reliable, rapid, and cost-effective quantitation of at least 6 targets from a single blood sample. This study is ongoing, so I'll be presenting some data today showing the performance of the 2 panels on a limited number of patient liquid biopsy samples. This slide shows the design of the 6-color breast cancer panel designed to monitor 3 PIK3CA mutations, HER2 amplification, and assess chromosome 17 polysomy. We also included an exogenous positive control to monitor for PCR efficiency. During the assay characterization phase, we used commercial DNA in order to set up the assay. Separability is a very good indicator of whether you are getting clean and specific amplification for each of your targets. As you can see here in these 1D dot plots, the separability between the positive and negative droplets is very high for each target. And good separability is crucial for correct and reliable copy number determination. You can also visualize the separability of the targets in a 2D dot plot format. This is a useful way to view data in a multiplex assay where you can easily visualize the double negative population, the single positive populations, as well as double positive populations for each color or target combination. We also examined the linearity of the assays and confirmed a high linearity with R-squared values approaching 1. Here's data showing 2 of the PIK3CA mutations. We then analyzed the limit of blank for each of the PIK3CA mutations, which was found to be 6 droplets. We also analyzed the limit of detection, which was very low at 0.8 copies per microliter for each of the PIK3CA mutations. And as I previously mentioned, linearity scores approaching 1 for all 3 PIK3CA mutations, as well as the ratio of HER2 to TSN control. This data indicates high performance of the 6-color panel in crystal digital PCR. However, this work was all performed using commercial DNA. Therefore, we next needed to test the panel on patient samples using the workflow shown here. We took whole blood samples and extracted nucleic acid from the plasma. We then made our PCR mix, loaded our 25 microliters into the Sapphire chip, and performed our 6-color crystal digital PCR using the breast cancer panel previously described. We were excited to see a perfect correlation between the experimental detection and theoretical presence of PIK3CA mutations, HER2 amplification, as well as chromosome 17 polysomy using the 6-color digital PCR panel. There was one exception in the PIK3CA data in sample 4, and this was found to be due to the fact that the presence of the mutation was below the limit of detection. We also designed a rectal cancer crystal digital PCR panel shown here to detect the presence of 4 PIK3CA mutations and an exogenous positive control to monitor PCR efficiency. As with the breast cancer panel, we first performed assay characterization by visualizing the performance of each assay by 1D dot plots. As you can see, the separability scores are very high and thresholds easily placed between the positive and negative droplets. Again, separability can be viewed and confirmed by 2D dot plots as well. As with the breast cancer panel, we measured the limit of blank, the limit of detection, and the linearity of the rectal cancer panel. We found a low limit of blank for each of the 4 PIK3CA mutations, as well as a low limit of detection ranging from 0.65 copies per microliter to 0.77 copies per microliter for all 4 PIK3CA mutations. And finally, linearity scores approaching 1 for all PIK3CA mutations. Once again, we confirmed our results using DNA extracted from patient plasma samples and performed the 6-color crystal digital PCR workflow. Results using 6 patient samples showed that all PIK3CA mutation concentrations measured by 6-color crystal digital PCR correlated well with the theoretical concentration. In conclusion, we have shown that 6-color crystal digital PCR is compatible with a variety of sample types. We have also demonstrated that combining crystal digital PCR with non-invasive liquid biopsy samples can be used to screen for and monitor disease progression with high accuracy, sensitivity, and precision. The 3 multiplex oncology panels for lung, breast, and rectal cancer each demonstrated high clinical utility. And overall, 6-color crystal digital PCR can be used to maximize information output of your precious samples while minimizing cost and time to results. There's one more live webinar to round out our 6-color crystal digital PCR series. This webinar is June 30th on the benefits of crystal digital PCR for environmental and food testing. You can register on our website, where you can also find a recording of the June 11th webinar in case you missed it. And I'd like to thank you for your time and attention, and I invite you to visit our website at stillatechnologies.com for more information about the NICA system and workflow, including application and technical notes. You can also find us on LinkedIn, Twitter, and YouTube. And if you'd like to learn more about digital PCR, please visit genepy.com, where you can find many useful tools to help you set up a digital PCR experiment, as well as how to help you analyze data. And with that, we're happy to answer any of your questions.
Yes, thank you very much, Kim, for that very nice presentation and the great data sets that you presented. And as you say, let's get ready and start with the question section with our first question, that is the threshold position automated, or do I have to do it manually in the system?
Yeah, the threshold is actually automatically set by the Crystal Miner software. That's one of the really great features of our fully integrated software.
Okay, thank you very much. So the next question will be, how do you determine the total droplet count?
That's a great question. Determining total droplet count is really important in order to have an accurate Poisson statistics. So we actually incorporate the use of fluorescein for our probe-based assays. And the fluorescein will be able to detect every single droplet and give you an accurate count of total droplets.
Awesome. Okay, thank you very much. Next question would be, how many samples can you run at a time?
So with our Sapphire chip, it's a total of 12 samples. So it's 4 samples per chip, and you can load 3 chips at a time.
Okay. Then a question fitting right after in is, what would be the dynamic range of the system?
Yeah, the dynamic range with the Sapphire chip is 5 logs. It ranges from 0.2 copies to 20,000 copies per microliter.
Okay, great. So, can you estimate how long does a scanning take on our NICA system?
Yeah, on the 3-color system, the scanning takes about 10 minutes, so it's 50 seconds per chamber with the current 3-color system.
Okay. Then the question would be, running on a multiplex as it is possible with your system, how much work is it done, or can we use already provided assays from other companies?
That's a great question. For other companies, I believe if the fluorophore would fit within the channel, it's definitely worth a try. There may need to be some tweaking to the system as with any assay, but I think that's to be expected with any experiment. But as long as the designed assay with the attached fluorophore fits within the emission and excitation spectrum, it should work just fine.
Okay, so we can run quite a lot of assays on the systems. One question would be, do I have to decide on the 3 or the 6 color system? So do you have both available?
Currently, right now, the 3-color system is what's commercially available. We are taking orders for the 6-color system, but it should be delivered by the end of the year.
Okay, thank you very much. So right now, and I've noticed that because we had to change the link this morning, unfortunately, some people could not join the webinar as planned. And I think we have a little bit less question than expected. So I would say we can close the session a little bit early. For the ones who have attended the webinar, this webinar will be recorded and be placed after on our webpage. And we will, of course, contact all the people that unfortunately could not join that nice presentation with Kimberley and me. So also they will get informed. And for any question, please contact us on support@stilla.fr. Thank you very much for everybody joining our web session.